My name is Dean Follman. I'm head of the Biostatistics Research Branch at NIAID, and I'd like to welcome you to the fifth presentation in this new lecture series from the COVID-19 Scientific Interest Group. To start off, I have a couple items to share. We are now offering CME credit for this lecture series, and the code for today is 24876. That's 24876, and we'll repeat it again at the end of the lecture. Next Wednesday, we're delighted to have Dr. Tara Palmore, the hospital epidemiologist at the Clinical Center. The title of her lecture is going to be Sailing Close to the Breeze, Hospital Epidemiology in the COVID-19 Pandemic. If you have questions during today's talk, you can use the question form on the bottom of the videocast webpage. This is at the bottom of the very webpage you are using now to watch the lecture. You can look for and click on the words live feedback form, which will take you to a new web page and a form to submit your question. Questions will go directly to the host, which is Irene Soretti, and she'll relay them to the speaker at the end of the lecture. And please use this form to submit questions to the host. This lecture is being video recorded and will be available tomorrow or Friday. And with that as bookkeeping, I'd like to introduce Dr. Lori Dodd. Dr. Dodd is a mathematical statistician at the National Institute of Allergy and Infectious Diseases. She grew up in the West and she studied anthropology at the University of Utah. And as part of her work there, she ended up working with the Navajo Nation, particularly the elders there, to try and understand how they were adjusting to change that was happening around them and how their culture was being affected by that. This work that she did was actually of local interest and ended up being in a newspaper article that was written about her. And if you go into her office today, you'll see that newspaper article on her wall, and the title is God on a Horse, and it goes into some detail about the work she had done there and some of her findings. So if you're in your office, you should definitely look at that. She didn't stay in anthropology, though. She ended up going to Harvard to study statistics for a while, and then it ended up at the University of Washington to get her PhD in biostatistics. After graduating, she worked at the National Cancer Institute for about 8 years, and then finally we were able to entice her to come to NIAID. We're very happy about that. She has interest in clinical trials, emerging infectious diseases, tuberculosis, and diagnostic testing, and she's published and written and lectured extensively in these areas. And as of today, I think of noon or maybe 9 a.m., she's officially head of a new clinical trials research section, which is devoted to running clinical trials, especially in outbreak settings. I've seen her build this group, and she's hired excellent people throughout. This group has a great sense of esprit de corps and loyalty, and they function great under pressure. She's received the Department of Health and Human the DHHS Secretary's Award for her work in the West African Ebola outbreak. And recently, the PALM trial, which we'll talk about today, won the Trial of the Year for 2019 from the Society for Clinical Trials. She was lead statistician for both of those studies and was deeply involved, along with her group, running those trials. We sit close together, well, not now, but on running trials in these outbreaks, I've seen firsthand, there's stresses and stuff happening and so on. And Lori has a certain grace and resolve that allows her to sort of keep moving forward, even though there's a lot of stuff happening and that can be daunting, I think, to some, but not her. So I think NIAID's really lucky to have her, and I'm proud that she's part of us. And with that, I'd like to introduce Dr. Lori Dodd.
Thank you, Dean. Can everyone hear me? I mean, not everybody, I think, can respond, but can you hear me, Dean?
Yeah, I hear you great, Lori.
Okay. So I was asked to give a talk about my experiences on conducting clinical trials in public health emergencies, and I want to reflect on my experiences from 2 Ebola outbreaks and my recent experience on the COVID ACT trial of remdesivir. Let me see if I can get my slides to advance. So I have 5 themes that I want to go through. The first is that randomization is an essential scientific tool. Trials need to start quickly. You don't want to miss the epidemic wave, but at the same time, you need to avoid recklessness. Enrollment is always an issue. You either have too few or too many, and both create challenges. You need to plan for uncertainties, but recognize you cannot plan for everything. And then finally, I want to emphasize that randomized trials should be seen as an integrated part of an outbreak response. So to highlight the 3 studies that I've worked on, the first is Prevail II, which was conducted in 2014 to 2016 during the Ebola outbreak in West Africa. It was a randomized controlled trial of ZMapp versus standard of care. The second trial is what we refer to as the PALM study, which was another study on Ebola conducted in the Democratic Republic of the Congo in 2018 through 2019. This was a 4-arm trial that was randomized to either MAB114 or Regeneron, this drug NEB3, which are monoclonal antibodies, and remdesivir, versus ZMapp, which was considered the standard of care based on the Prevail II study from the West African outbreak. And then finally, I want to reflect on my recent experience with the ACT study, the adaptive COVID-19 treatment trial, which is an adaptive platform randomized controlled trial, with the first ACT, which we call Act 1, being a randomization to remdesivir versus a placebo for remdesivir, which is now completed. We've started Act 2, which is a randomized controlled trial of remdesivir plus baricitinib, an immunomodulator, versus remdesivir plus a placebo for baricitinib. So I want to think back to 2014. At that time, there was much controversy over randomization. And you can see from a few articles, I wish I'd kept more, but there was a lot of news about whether randomized controlled trials could be conducted at all. And the debate ranged from it being unethical in the context of an outbreak to it being the ethical thing to do as the best way to establish treatment efficacy during an outbreak. And so as a result, there were many trials conducted, and only one of them was a randomized controlled trial. So here, I don't know if people can see it on their screens at home, but this is from a meta-analysis. It shows the 8 different trials that were started, and the enrollment dates ranged from starting in June of 2014 for some of the control groups, to our randomized control trial, which started in May of 2015. I've highlighted that these are all non-randomized controlled trials, which created a lot of challenges because there's a lot of sources of patient heterogeneity. There's always sources of patient heterogeneity in any of the diseases we study, but in particular with a novel infectious disease, there are a lot more things, at least I think, a lot more sources of heterogeneity, and this really can make confusion about interpreting potential treatment efficacy when you're comparing a study of one drug versus some kind of historical control. So in the Prevail setting, we learned that there were different diagnostic platforms that made a big difference. Viral load and infection is a factor in terms of prognosis, and the estimates of how much viral load there is can vary by the diagnostic platform. Things like age or sex can influence prognosis, time from illness onset to treatment, comorbidities, differences in supportive care by center and over time, and then there's always in the background questions about whether the virus is changing and if this is somehow affecting improved outcomes or making outcomes worse. What's interesting to me is I pulled this from a previous slide, and I think these sources of heterogeneity are every bit as relevant today as we embark upon studies for COVID treatments. But to my point about getting started early, this is a plot of the epidemic curves from West Africa, Guinea, Liberia, and Sierra Leone. So those are the jagged colored lines, and the bars show the studies that were started. So the scale for the epidemic curve ranges from 0 to 700 on the left-hand side of the plot, and on the right-hand side, the scale ranges from 0 to up to 14 for the number of trials. So the scales are very different. But what you can see, importantly, is the trial started quite late in the course of the epidemic, and the Prevail II study started especially late. The challenges, there were many, and I didn't provide an exhaustive list here, but there was the design debate, there were ethics approvals, they're getting pharma on board, international coordination, supplies, staffing, to just name a few. Some of these things can be overcome. I think that the design debate has gone quite far, and so those timelines can be improved. Ethics approvals have improved, those timelines certainly. But there are certain things that are just difficult, and you can't shorten the timeline completely. So in Prevail II, we enrolled 72. We targeted 200, and the outbreak ended. And so we were left with suggestive evidence that ZMapp worked, but it didn't cross the standard evidence of statistical significance. And unfortunately, this was, in the context of having seen over 28,000 cases of Ebola with 11,000 deaths. And in the context of this study, or this outbreak, there were at least 8 studies of treatments launched, 8 with a control group that ended up being published. And Prevail II was the only randomized controlled trial which ended prematurely due to the outbreak end. So the outbreak ended without rigorous evidence supporting treatment efficacy. So in the interim, the National Academy of Medicine conducted an investigation and on the experience with Ebola in West Africa with respect to clinical research. And they published this report in 2017. And their conclusions were that the core principles of science and ethics in conducting clinical research do not and should not change in the midst of an epidemic. They concluded that the randomized controlled trial is ethical and the most appropriate study design to use because it's the most efficient and reliable way to determine whether an investigational treatment or vaccine is safe and effective. And clinical research studies must have scientific and social value, respect for and engagement with people in the affected communities. And then also it's important to have post-trial access to any investigational agent that proves effective. So now we're going to move to 2018 in the Democratic Republic of the Congo with their outbreak in North Kivu, Ituri province. And there was also some evolution in terms of the standard of care for the Ebola patients. So you can see here the new pods to treat patients are being utilized. Unfortunately, though, the case fatality rate was still quite high, about 67%. There was no approved treatment for Ebola. And so this was the environment we walked into to work with our Congolese partners at the Institute of the National Biomedical Research, or the INRB, in the Congo, to start what we call the PALM study, which is Pamoja Tulinde Maisha. I'm sure I'm not saying that properly, Olivier, if you're out there, which means, it's a Swahili word, which means together save lives. So this was the study design. We had a 4-arm trial, and I was quite concerned about having a 4-arm trial because of the Prevail II experience, and you never know if you're going to have enough cases to enroll to fully to any study, let alone a study with 4 arms. The drugs were ZMapp, MAB114, remdesivir, and Regeneron. You'll note here that we had this little purple square to indicate the possibility that there was a drug shortage. That's always a concern, or often a concern, if you may run out of supply of a drug in a setting like this. And so we had to make sure that we had a protocol plan for that, and our plan was to allow for that shortage to happen. And then in the analysis, we would only evaluate treatment effects for subjects who were enrolled concurrently. The primary outcome was 28-day mortality. In this case, here's a picture of the team from NIAID. We're in our hotel, which became home for a while in Kinshasa, and we had just gotten our ethics approval. And so in this setting, we started, the first time Cliff Lane called me to talk about this was, I think, around August 8th. And then by November 20th of 2018, we'd actually gotten IRB approval and we're ready to start. So what does that look like? So this is the WHO epidemic curve. And you can see that in this case, we were able to catch the cases quite nicely and enroll quite rigorously. So in Prevail II, it was 7 months from the concept to the start of the study, while in Palm, it was 3.5 months. And that made a huge difference. This is an environment where days and minutes matter a lot, which means you don't sleep much. So just to focus on enrollment a bit more, Prevail II, as I said, the enrollment ended when the outbreak ended with only 72 total, but this was not the case with Palm. We ended up enrolling up 681 in the main phase of the study. And the pace of enrollment was quite quick. So we enrolled, within just over like two months, we enrolled 400 participants, which seemed like a lot at the time. It was certainly a major accomplishment for the entire team. On August 9th, we had a DSMB meeting, and at that time, there were 681 people enrolled. Of those 681 people, not all of them had made it to the day 28 time point. So there were 305 subjects who hadn't made it to day 28, which is important when you start thinking about how you will be able to inform the outbreak about treatment decisions, efficacy, to make statements about efficacy. It takes time for data to come in, and this can be very uncomfortable in the setting of an outbreak. So another comment about managing rapid enrollment, more patients means busier staff, it means more supplies are needed, it means more data that has to be managed and queried. All these things take a lot of effort. It also means more data to inform interim analysis results. So it means that you can make faster decisions about treatment efficacy in theory. But as I pointed out on the previous slide, data take time to mature. So if you enroll a patient today in a study, then his or her 28-day results are probably not available for another 5 weeks. They have to reach that 28-day time point. It has to be entered into the system. There have to be data checks. All of that can be compressed to a certain extent, but when you're getting overloaded with patients, the ability to do that quickly, it takes more and more people to keep up. And actually, we rely quite heavily on the field teams to respond to those queries, and they're very busy responding to the outbreak. So it can be an anxiety-producing time period. I want to make one comment about interim monitoring, since I expect not everybody's familiar with interim monitoring. So in clinical trials, we monitor for early evidence or harm. It's important that we do this so that we can make decisions early about efficacy or harm and notify the study team and make changes if there are definitive evidence early. But the problem is that early data are unreliable and early trends can reverse. There are many examples of those, and I'm not going to go into that in this talk. But interim monitoring uses formal statistical procedures to do this. Trials are monitored at a few interim analyses, which we often refer to as looks, rather than looking at the data continuously. Between the interim analyses, the groups of data accumulate, so you may also hear the term group sequential monitoring. And that's how interim decisions are often made. However, a data safety monitoring board uses this as one of many other pieces of evidence when they make recommendations. the DSMB will consider the totality of the evidence when making their recommendations. And I will say that the decisions about their recommendations are rarely easy. We're very lucky to have data safety monitoring boards and we rely on them quite heavily for their advice. So in the context of PALM, I pulled this from a previous timeline. We had data safety monitoring meetings. I'm just referring to the 2019 timeline. We had multiple of them. So I want to focus on this last event. So between July 8th and August 9th, we had a major uptick in enrollment. And by August 9th, we were not at full enrollment, but we had reached 681 enrollees. As I've already said, the time from enrollment until the 28-day mortality results were in the database could be 5 weeks, in some cases 6 weeks. So at that time point on August 9th, only 376 patients had a day 28 outcome in the DSMB report, which in statistical terms, that's only about 55% of the total study information. This is sort of, juxtaposed against the fact that the study was nearly 94% complete. And so this, the DSMB is really focused on the amount of data that has reached maturity in many cases. And so this creates attention. I was quite concerned. But fortunately, our DSMB was wise, and early on they had realized that looking at deaths by day 10, they could predict the deaths by day 28. So in Ebola, most of the deaths occur early. And as a result, the DSMB realized that they could look at much more data than just the 55% of the total study information, and in making their recommendations, because looking at the day 10 mortality results were highly predictive of what the day 28 results would predict. And again, there's a responsibility to make recommendations about treatment efficacy as quickly as possible, because patients are dying, and if you can offer them treatments that will save their lives, you have an obligation to do that. So here is an example of the interim monitoring for PALM. And these lines, the blue lines and the red lines, correspond to what we call stopping boundaries. And so here you can see that this particular RMD was better based on the surrogate. It had clearly crossed the boundary. So this was one of many pieces of evidence that was presented to the Data Safety Monitoring Board, not by me because I was a blinded statistician, but by the unblinded statistical team. This was presented to the Data Safety Monitoring Board, and they deliberated. And they recommended that the study stop on August 9th because they concluded that MAB 114 and Regeneron lowered mortality relative to ZMapp. So things got busy quite quickly after that. The site investigators were immediately informed so that patients who could be offered MAB 114 or Regeneron. And also there was a lot of coordination so that all Ebola patients in the DRC could be offered MAB 114 or Regeneron. And on August 12th, there was a press release from NIH that was convened by Dr. Fauci and Professor Muyembe, who's the head of the INRB. Ultimately, the results were published in the New England Journal, and this is from the final study report from December 12th of 2019. And you can see in the middle here, there are the survival curves with MAB 114 and Regeneron at the bottom, and remdesivir and ZMapp at the top. And there's a clear separation indicating an improvement in mortality for Regeneron and MAB 114. So now I want to turn to COVID and our ACT trial. The this is intended to be a platform study that arms can be added or dropped in some pretty structured ways. And Act 1, as I indicated, is the first stage of this ACT. And it's randomization to placebo versus remdesivir. Act 2 is a randomization to remdesivir plus a baricitinib placebo versus remdesivir plus baricitinib. And it includes patients hospitalized with COVID-19 with some evidence of lower respiratory tract disease. So the timelines here are even more aggressive than the previous PALM study. The study concept began on January 1st. And you can see from the slides, the synopsis was started on the 6th of February. The IRB approval came on the 20th of February, and then on the 21st of February, the first site was activated. This was at a time when the majority of the cases were still in China. And there was very little information that was known about COVID disease at that time. And so this led to a lot of challenges for designing a study for COVID. Unlike Ebola, where there had been 10 outbreaks in the Congo and more knowledge at the time, COVID was a novel disease, and we were learning a lot about it at the time of developing the protocol, and we continue to learn a lot more about this disease. So this, as I said, was less than 1 month from concept to study start. So one of the big challenges that we faced was what the primary endpoint should be. Little was known about the course of COVID-19 when the trial was designed. The length of illness, was it 1 week, 2 weeks, 4 weeks, 6 weeks? Initially, we thought it was shorter. And as more data came in, we learned that some patients had a much more prolonged course of illness. The WHO initially published an ordinal scale for evaluating efficacy. And this is a modified version of that we used for the protocol. And it ranges from death to not hospitalized and no limitation on activities, with the categories in between primarily relating to oxygen requirements, with the category 7 being hospitalized on invasive mechanical ventilation or ECMO. And then each step down from there indicates a lower requirement. This is about as statistical as I'm going to get, I think. So I want to talk a little bit about how to analyze these data. So on the left side is a stacked bar plot of the ordinal scale for lopinavir-ritonavir, which was the first Chinese randomized controlled trial to report out. And you can see that the different shades correspond to progressively worse categories on that ordinal scale. So the left-hand side is the lopinavir-ritonavir, and the right-hand side is the control arm. And what you want to see is that there's more of the lighter colors in your treatment arm, indicating a shift in the distribution between the treatment and control. So if you're testing those categories, the different ordinal scores, one way to do that is to use what's called a proportional odds model, and it's just one way of testing the shift in the distribution. The issue is that has to occur at a fixed time point, at least without doing some more advanced methods. So that was the initial proposal, was to use a day 14 proportional odds model. An alternative is to use a time to event endpoint, such as time to recovery. And so on the right-hand side, I've plotted the Kaplan-Meier curve from that same lopinavir-ritonavir study that looks at a time to improvement. And you can see, actually one thing you can see is my day 14 got misaligned. this line over here. I don't know if you guys can see my mouse, probably not, but this line here, the blue line with day 14 above it, is clearly in the wrong location. So anyway, you can see that this time to event then is capturing more of the time course of illness. And in a disease like this one, in which there can be a range from sickness from, ranging from a week or so to 6 to 8 weeks, it seems important to capture the time course. And so that was, we ended up using a time to recovery endpoint. Just one other thing to note, that, the analysis method, as I've just indicated, this is saying in words, corresponding to the shifts in the full scale, is the proportional odds model at a fixed day. So in our case, it was 14 days after randomization. There are some that argue that this proportional odds approach is the most efficient. And by most efficient, I mean that the answer can be obtained with the fewest number of subjects. And so we've countered to that because this is not always true. And so that was, that's an ongoing debate. And we've just submitted a paper arguing this, that we think that the time to event has some serious advantages. Just to emphasize and conclude on this point, there was growing concern that the illness course was more protracted than previously thought. We're learning that there was a heterogeneous disease manifestation with a time course ranging from a few days in the mild patients to 6 to 8 weeks in the critical patients or death. And we were concerned that the risks of a single time point for evaluating treatment effect would mean we could miss the treatment effect, because we did some modeling that revealed that meaningful treatment effects could be missed if the wrong day was selected. Also, we felt that the time course was an important aspect of the disease, and we wanted to capture that. So on March 21st, we discussed this. I presented this on a protocol team call, and the decision, was made to make that change. There were 77 subjects enrolled at the time. That decision was made without looking at any data. And so we changed to the time to recovery, or in other words, the time to being well enough for discharge over 28 days. And importantly, there were also no data collection requirement changes. We were already collecting the ordinal scale, and it was really just going back to these bottom 3 categories of the ordinal scale. So these 3 categories refer to what we refer to as recovery. And one thing to note, this middle, or this 3rd category here that says hospitalized, not requiring supplemental oxygen and no longer requires ongoing medical care, was introduced early on during the planning, as we were, as patients were coming from the cruise ships, and they couldn't return home because they were sent to medical centers far from home, and they were kept in the hospital longer than they might have been otherwise, either for infection control purposes or until they could get sent home. So we felt like we needed to capture that as a category because those patients were in relatively good health and they didn't need to be hospitalized. They just were kept in the hospital for other reasons other than illness. Okay, so the ACT study was impressive in terms of the number of sites that we were able to bring on. And this map here shows you how many sites were included. So we had multiple sites in the US. We had South Korea, Japan, Mexico, Greece, the United Kingdom, Germany, Denmark, Spain, and Singapore. And I think even the logistics of managing a weekly PI call is quite impressive. And as a result, the trial enrolled it very robustly. And I will point out that I think this curve here, so on the bottom curve, you can see the enrollment from Act 1. And so again, it started on February 21st. And then by April 20th, there were 1,063 subjects enrolled. On the top panel, you'll see the timeline for the epidemic in the US, and you can see that it mapped quite nicely. I can tell you that looking at this curve and having experienced this curve, it looks a lot smoother than it was. I mean, this amount of enrollment to get this up and running took a massive team effort from lots of people. And so it's a testament to the whole team that was involved in this. So on Monday of the 27th of April, the DSMB met for what I thought was going to be an interim efficacy analysis. There was a plan to do an interim analysis based on 200 recoveries. This is an event-driven study, so it's not fundamentally designed to enroll a total number of subjects. It's intended to enroll based on the number of events. So the study was designed to enroll subjects to give 400 recoveries, which is how the interim analysis timelines are planned. So the interim analysis was planned for 200 recoveries, but because of the massive pace of enrollment, the data set that was reviewed exceeded the total final sample size, which was intended to be 400, and in fact, it included 482 recoveries. So in that sense, the study had met its objective of enrollment, and this was the primary analysis. The DSMB recommended that NIAID be unblinded to the primary analysis and key secondary analyses to determine public health implications. And the primary outcome results had crossed the boundary of statistical significance. Furthermore, there was an obligation to inform the study team and offer remdesivir to those still on study, those in particular who were on placebo. If they might receive benefit, there was an obligation to offer it to them. So on the 29th of April, NIH released, NIAID released a press release that indicated that based on their review of the data, they noted that remdesivir was better than placebo from the perspective of the primary endpoint, which was time to recovery. The preliminary results indicated that those who had received remdesivir had a 31% faster time to recovery relative to those who received placebo, with a p-value less than 0.001. And specifically, the median time to recovery was 11 days for patients treated with remdesivir compared to 15 days for those who received placebo. Soon after, the FDA issued an emergency use authorization on May 1st, and this is in effect. The ACT preliminary results, the study is still in follow-up, so I want to emphasize that the specific numbers will change. We do not expect the conclusions to change, but as more data become mature, then there will be slight changes in the numbers. Right now, as of April 28th, about 70% of the subjects had a primary outcome data available, and about 30% of participants had not met the primary endpoint. Site monitoring is ongoing, and the data are not fully clean. But this is an example. This is from the data, a Kaplan-Meier plot of the time to recovery for remdesivir to placebo. And you can see here, again, I wish I had a pointer. At this 50% proportion recovered, you can go to the right and figure out what the median recovery time is from this plot. So this is the primary analysis, which corresponds to a p-value of less than 0.001. Again, so the preliminary data supports that patients who received remdesivir had a 31% faster time to recovery. It was 11 days versus 15 days. There was a possible decrease in the mortality rate. The ACT study needs to be completed, the data need to be finalized, and the manuscript needs to be peer reviewed. As I've said, we don't anticipate significant changes in the findings. While remdesivir is a significant improvement for treatment of COVID-19, additional studies are needed to continue to decrease the morbidity and mortality associated with this disease. And then finally, I want to say that Act 2 started on Friday. So just to conclude with the 5 themes I wanted to go through. Randomization is an essential scientific tool. Heterogeneity has many sources. Diagnostic tests, again, we learned this in Prevail II in the West African Ebola setting. This is also true today. The dose of infection, and by that I mean how much virus one gets upon infection, may play an important role in terms of how sick one gets. We don't know that here, but certainly that's been speculation in the Ebola setting. Patient characteristics play an important role: age, sex, and comorbidities. The standard of care can change, and it's certainly changing with COVID. The duration from onset of illness to care can play an important role. And sometimes this can play a counterintuitive role. So in the Ebola setting in Prevail II, one of the things we noted was that some patients who show up late for care in the Ebola setting may tend to do better, and that's because there's something called a survivor bias. If you survive long enough to make it to the hospital, then chances are you're going to do well. That was true in the Ebola setting, not true here probably. Hospital capacity is also an issue in all of these settings, not to mention the staff, the dedicated nurses and doctors and care providers who, are just over capacity, working hard to keep up, not only with patient care, but also then with research demands. The Prevail experience confirmed the value of randomization, and the value of randomization is quite recognized now. We're certainly not having the same debates we had in the West African era. And I think the lack of that debate has helped us get trials started more quickly. I think there's also a lot that has been learned in terms of operationalizing trials and getting things up and going. So in Prevail II, it was 7 months from the concept to start of study. In Palm, it was 3.5 months from concept to start. And in ACT, it was less than a month from concept to start. Enrollment is always an issue. It's either too few or too many. In Prevail II, the study ended before full enrollment, and we were stuck with results that were suggestive about ZMapp and not definitive. And that set us up for some debates when we started Palm about what the control arm should be. Ultimately decided that ZMapp didn't look harmful, and so that made the decision to have ZMapp as the control arm. In Palm, the rapid enrollment made for uncomfortable delays in interim analysis timelines. In that setting, we were fortunate to have a surrogate. 10-day mortality was highly predictive of 28-day mortality, and that made results available faster, and we were able to stop the study sooner. In Act 1, we enrolled the full study and beyond in about 2 months. There's no such surrogate here as there is in Ebola. And then also, because we enrolled so quickly, definitive conclusions were reached before the full enrollment cohort had reached their final study visit. Finally, the last 2 points. One is to plan for uncertainties, but recognize you cannot plan for everything. Novel infectious diseases, by definition, there's little known about them. So you have to expect that unexpected things will happen and anticipate what you can, but be prepared to change. But you have to do this without sacrificing scientific standards. One thing I haven't talked about, but could, is there are limits to how adaptive trials can be and maintain the scientific integrity. So it's important to anticipate and plan as much as possible, but not sacrifice all the standards. And also, I think one thing you can, spend a lot of time trying to anticipate every scenario, but ultimately, what you need to do at some point, you need to just be prepared for whatever unexpected thing that's going to happen, and have, your experts at near and by so that you can consult with them and make quick decisions in light of this unexpected thing happening. Finally, randomized controlled trials should be an integrated part of the outbreak response. So PALM and ACT are both examples of how treatment advances that occurred during the course of an outbreak can inform treatment decisions for infected patients during an ongoing outbreak. But importantly, rapid communication of reliable results is paramount. And so we rely on the statistical machinery of interim monitoring and a data safety monitoring board to make these recommendations and inform the study team about results when they become reliable, and then it's up to the study sponsor to communicate them. So I want to conclude with a few credit slides. I'd like to thank the multinational Prevail II study team. And I would also like to thank the PALM study team. It was a large group as well. And finally, the ACT study team. So with that, I will stop. And Irini, I can take questions.
Thank you very much, Lori. Can you hear me well? This was fantastic.
Yes,
Thank you for the presentation. So I just wanted, before we start with the questions, I just wanted to remind people that the CME code for today is 24876. And for those who still want to send questions in, although I think we have a pretty full list of questions, that you can do that from the question form on the videocast webpage, which is at the bottom of the webpage you're watching, and look at the live feedback form that will take you to a new webpage and a form to submit your question. So the first question is, thank you for your outstanding talk, and whether you could comment on the importance of research preparedness, because certainly it takes different time for clinical trials to get off the ground. For example, in Liberia in the beginning, the Ebola was slow. In Brazil, for the Zika, it was faster versus China, which was very fast for COVID. And whether in your experience, is the baseline clinical trial capacity a critical factor, or is the protocol development and approval the rate limiting step? And then can capacity be rapidly developed on the fly? That's from the Deputy Director of the Fogarty Center.
Yeah, so there are multiple parts to that question, right? So one is the protocol development, clinical trials capacity. They're all important, and it's important to have a clinical trials or team to be able to rapidly get trials up and going, and you need a team that's experienced, right? So I think that was one of the important lessons from Prevail II, and that's where I got started in this, as well as many people. We learned a lot from that experience, and that's one reason why we were able to move so rapidly with the PALM study. I think in terms of protocol development, yeah, one of the things you have to make some decisions pretty quickly to get it moving, and you can shorten that timeline pretty quickly. But the rate limiting step, I think, is getting all the partnerships and things set up and getting going.
Great, thank you. So the next question is whether you could comment on the use of response adaptive randomization in the current pandemic context.
Yeah, so I'm not a huge fan of response adaptive randomization for lots of reasons, and maybe even the simple reason that if you think about the ACT study and how long it takes to get endpoints in, or let's think about the PALM study. By the time you get endpoints in, so the response adaptive randomization is a tool to change the randomization probabilities based on incoming data. There are many risks associated with doing that. So I'm not going to comment on that. I agree with the risks, and I have lots of concerns about those risks. But I think just from an operational perspective, getting the data in a reliable way, in a timely way, is pretty impractical. So you have to wait for those endpoints to come in, and then you have to adapt your randomization probabilities. And the logistics of doing that, I think, make it a very it's a challenging approach to use, and I wouldn't recommend it because I think there's lots of risks associated with it for little to no gain, and perhaps even more importantly, there's a lot of questions that can be raised about the study in the end. You know, clinical trials are meant to resolve controversy, not stir it. So my personal point of view is that we should stick to some tried and true methods.
Thank you, Laurie. The other question is whether you can comment if the treated patients were from the exact same post-infection stage or not, if there was any stratification based on that. There are two actually questions on that. The possibility that remdesivir actually may be more effective if it's given during an earlier infection stage. And I think that was suggested also by the Lancet remdesivir paper from China.
Yeah, so I think we'll have to wait and see when the final data are available. We did stratify according to baseline disease severity. We used the definitions that were used in China during their studies, roughly, and so we'll be able to get more clarity on that soon.
Okay, the other question, which I presume probably you don't have full data for, would be if there are any specifics you can share about age or sex and the outcome in Act 1.
Yeah, so the results, Irene, are still in preparation. We're working to get those out as quickly as possible. So they should be out there soon.
Okay.
I don't have anything I can say right now.
Okay. Could you perhaps comment on how the different sites were originally selected for participation in ACT and whether the same criteria would be used going forward?
I'm sorry, I'm not the right person to answer that question. Can I play
Okay.
the biostatistician card?
Okay.
I wish I could. I just don't.
No problem. I have a very nice question for you. Somebody is wondering if you could speak how your training as an anthropologist shaped your career in these emergent pandemics.
How long do you want me to talk? Well, so I think you need to be open and listen to people. And I think often in these settings, you're working in a multicultural environment. And you need, in order to do this successfully, you need to be able to, you need to at least try to understand things from different cultural perspectives. And so I do see that thread through my work.
All right. Let me see. Do you happen to know, I guess from the sites, you don't have any insight about the different sites participating in the study you mentioned. Is there somewhere where people can see the sites participating in Act 1? Is it on clinicaltrials.gov maybe?
I should know that, but I don't.
I would say probably
Yeah.
it should be there. The other question is whether you could comment on how death was handled as a competing risk to time to recovery analysis in ACT. And this, I don't know. I don't know this model, but they're asking whether you're okay with a sub-distribution model like the fine gray model.
So let me just start with this figure. So what happens with somebody who dies? So in a typical survival analysis, you're looking only at the time to death, which is the time to a bad event. When you're looking at time to recovery, you're looking at the time to a good event. And therefore, when somebody dies, you have to handle that death in a certain way. And so what we've done is to handle the deaths as censored at the last day of observation. So somebody who dies at day 9 is considered at risk of recovery, in essence, until day till the end of the observation period. Now that may make some people uncomfortable, but you can also think of this as giving somebody who dies an infinite recovery time. And we did a lot of simulations, and in fact, Dean's done a fair number of simulations, and this is part of the paper that we just submitted, looking at how sensitive this approach is to death. So we did some simulations where we assumed the recovery rates for the treatment and control arm were identical, and the only thing that differed was the mortality. And what we saw was that this approach did pick up a difference in the treatment arms under that scenario. So that made us feel comfortable. And there are some connections with the fine gray method, which maybe I won't go into now. But that's covered in the paper that hopefully will be out relatively soon.
Great, thank you, Laurie. The other question is how is basically the choice of the next step of the next drug? So how did you move to Act 2, for example? How did you choose a drug for that?
That's not my decision, so I can't really comment much on that, right? I mean, there was a deliberation and a process that was in place for that, and my role as a statistician was to evaluate how that would impact the study design and ultimately the sample size.
Okay. Do you foresee that similar adaptive designs could be used for vaccines, for example, for COVID-19?
Vaccines are different, and I'm in the sense that, you know, with a treatment trial, you want to keep, you know, you've got to keep treating patients. So we kind of want to keep the machinery rolling. And with vaccines, you may just vaccinate people and then wait for the events to come in. I don't know if Dean's on and wants to comment because he's more involved with vaccine studies, but I think it's an attractive platform, but I haven't thought through the implications for a vaccine study.
Okay, so I think we can wrap it up with this last question, unless Dean has any last comments. But I wanted to thank you again very much for a very enlightening talk. And I want to thank the audience and the people who send in the questions, and hope to see everybody again next week for Dr. Palmore. Thank you, Laurie.
Yeah, thanks, Irini. Have a good day.
You too. Bye-bye, everybody.